So hello, everyone. Thanks for the introduction. My name is Giacomo Marino. I'm a Bioinformatics Software Engineer in the Mayan Lab and a member of the Mount Sinai PGDAC team. And today I'll be presenting about Multiomics2Targets, which is a computational workflow to identify targets for cancer cohorts profiled with transcriptomics, proteomics, and phosphoproteomics. So just to begin, I'd like to note that Multiomics2Targets paper was recently published in Cell Reports Methods on August 9th, and there's an image of that on the left. And then on the right is the graphical abstract for the paper, which we show these various data types that it takes in, some of the tools that we utilize in the pipeline, some of the expected outputs, and then also the fact that this is an automatically generated report that can be shared and exported as a PDF. So just to begin with some background on a few of the tools that we utilize in Multiomics2Targets, the first is Target Ranger. And Target Ranger is a website and an algorithm that compares the expression of genes in uploaded tumor samples to the expression of those genes in several healthy human tissue and cell type atlases. So this includes processed data from ArchIS4, GTEx, HubMap, Tabula Sapiens, and others. And in the web interface here, we can filter these genes to prioritize those genes that encode for cell surface proteins. And we can also observe the expression of those proteins in that uploaded file versus the various healthy atlases. And this algorithm was inspired by a study from the Morris lab out of UPenn, in which they identified GPC2 as an oncoprotein and a candidate for immunotherapy in high-risk neuroblastoma. And the two criteria they looked at when trying to identify genes for this were those genes that were highly expressed in the tumor compared to normal tissues and cell types, in this case from GTEx, as well as those genes that encode for cell surface proteins. So GPC2 fit this criteria, and they were able to successfully develop an antibody drug conjugate, or ADC, that could target the GPC2 and selectively kill those cancer cells. So as part of the Multiomics2Targets paper and the pipeline, we systematically applied this algorithm to all of the 10 CPTAC3 cancer types, which involved first clustering these tumors into subtypes based on their RNA-seq expression, and then computing differential expression compared to two atlases, in this case, GTEx and ArchIS4, again, looking for those genes that were highly expressed in those tumor subtypes, and then lowly expressed across normal human cells and tissues. We then additionally apply a membrane filter for those genes that encode for membrane proteins, as well as annotate those targets with proteomic expression when available. So Target Ranger was published alongside its sister site, Gene Ranger, which takes in an individual gene symbol and shows the expression across a variety of transcriptomic as well as proteomic atlases. And this was published in Nucleic Acid Research in May of last year. Another tool that we utilize in the Multiomics2Targets pipeline is KIA3, which is a kinase enrichment analysis tool that relies on data integration from over 20 kinase substrate databases or data sources, which can be seen on the top right of the slide here. So these data sources have different amounts of coverage in terms of what kinases they have information for. And in the publication, we showed that taking a mean rank or utilizing as many databases as has information about certain kinase and taking the mean rank across those showed the best performance in recovering upstream kinases whose putative substrates are overrepresented in an input list of proteins or differentially phosphorylated proteins. So KIA3 is available as a web interface with some visualizations, as well as an API.
Hey, Giacomo, the KIA includes CPTAC data or just literature?
Sorry, what includes CPTAC data?
This tool, the KIA3, is that includes CPTAC data or just literature based?
No, so it doesn't include CPTAC data specifically, but these are some of in this top right figure here, these are some of the databases that it does include. So this is BioGrid, Reactome, a variety of phospho or kinase substrate databases, as well as those from literature. So it's a combination of many different sources.
Got it. Thank you.
Yep. A related tool that we utilize in the pipeline is Chia3, which is a transcription factor analysis, transcription factor enrichment analysis tool. And this additionally, this also relies on data integration of orthogonal omics. So here we also integrate various transcription factor target databases. And again, in the publication, we saw that a mean rank across these databases produces the most accurate ranking of transcription factors most likely to regulate that list of genes provided. So again, this is available as a web interface, and one of the visualizations is seen here on the left, as well as an API, which we utilize in the pipeline. So another related tool and algorithm is called expression to kinases, or X2K. And this infers cell signaling pathways from RNA-seq data. And this is done through looking at differentially expressed genes from RNA-seq and enriching for transcription factors with Chia3. And then inferring intermediates through protein-protein interactions from those transcription factors. And then finally using those intermediates and transcription factors to enrich for kinases that are most likely regulating that group. So in the end here, we get these inferred cell signaling pathways. So an example of which is seen on the slide here, and we've published on this a few times in the past. And this is also available as an Aperture in the Aperture catalog as well. So with the availability of matched RNA-seq and phosphoproteomic data from CPTAC, we wanted to revisit this algorithm and pipeline such that we can compare the kinases that are enriched directly from the up and down regulated phosphosites to those that we infer through the X2K pipeline from the RNA-seq data. So a few things different from what I was explaining before on the X2K is we're doing this per sample. So for each inputted sample, we're taking the top and bottom up and down regulated genes, enriching those with the Chia3 API, and then doing some rank normalization to account for literature bias as well as the gene space. And then we tested several different gene set expansion methods for those intermediates, including ArchES4 co-expression and protein-protein interactions through another integrated source called Genes to Networks that has many interactions from BioGrid and other sources. And then using those again with Chia3 to enrich for kinases, performing that same type of rank normalization and getting that list of ranked kinases per sample, and then comparing those to what we got from the phosphoproteomics. So this is part of the analysis that we do in the Multiomics2Targets workflow, as well as in the paper itself. So along with the analysis we did on the CPTAC3 cohort, we also developed the Multiomics2Targets site. And this is a customized Aperture, and Apertures are basically interactive Jupyter notebooks that provide HTML input forms that customize how the Jupyter notebook is executed. So here, in this case, you can upload the various data types, transcriptomics, proteomics, phosphoproteomics, along with metadata. And this is really geared towards the CPTAC data in general. And then there's a variety of parameters you can modify depending on how you want the pipeline to run, including the method for predicting intermediates, as well as selecting cell lines from DepMap for target validation. And then additionally, we have all 10 CPTAC3 cancer types canned and ready to load immediately so you can analyze that data. So importantly, we also understand that there's not always a great availability of all of these matched omics. So users can upload any combination, one or two of the omics, to receive a partial analysis report. So we plan to, and for any working group that we're involved in, we plan to run this as a very easy process. And it's also important to note that this is an open source project. And since all of the analysis is performed in a Jupyter notebook in Python, it's very easy to add additional analyses or apps that add to the workflow itself. And so this is all available in open source on GitHub as well. So this is the workflow diagram for Multiomics2Targets, and this utilizes many of those apps and background apps I was explaining before. So from the transcriptomics data on the sample level, we're performing that X2K analysis, as well as at the cluster level, performing target ranger analysis to identify those cell surface proteins that might be good targets for immunotherapy. We also additionally enrich the phosphoproteomics directly for those kinases, produce UMAPs, as well as using the proteomics to annotate those cell surface targets we identify from Target Ranger, as well as driver kinases we identify through X2K and from the phosphoproteomics. So now I'll just show a quick demo of the site. So here I'm just loading the default example for glioblastoma. And you'll see the report takes the form of a research publication with an abstract, introduction methods, and then a variety of results. So here are characterizations of various clusters through selected enricher libraries. We also have automatically created descriptions of the results that are generated with GPT-4 and put in yellow boxes. So those really explain the enrichment results there and appear in other places across the pipeline. So you can see there are a multitude of figures and tables produced in this analysis that can be investigated further. And these are all, the URL is constant and can be shared and is stored in an S3 bucket. And this whole report can be exported as a PDF and shared as well. In the future, we'd love to, through collaboration, test these predictions in the commercially available cell lines we identified, as well as patient-derived cell lines and in PDX models, and hopefully, again, with the help of collaborators, develop antibodies and ADCs. We'd also like to expand this methodology to single-cell multiomics profiling, and specifically looking to target perturbed or diseased cell types for removal, perhaps leveraging deconvolution analysis such as the Bayes de bulk that Francesca has been doing for many of the working groups. And then finally, also, we'd like to expand the methods to the Lynx L1000 connectivity mapping, which would allow us to identify reverse or mimicker compounds to drive cell populations to a healthier phenotype. And with that, I'd like to just thank everyone in the Mayan lab, as well as all members of the Mount Sinai Proteogenomic Data Analysis Center and Dr. Pei Wang's lab, as well as the NIH for their funding. And thank you all for listening.